Alrighty, we are on, what is it, 9.1 and 9.2. We're going to be learning about the cell cycle. So we've learned about cells. We know that cells are these little tiny dudes. They often have a nucleus, right? So why don't we, well, let's put it this way. When you were a little tiny baby versus how you are now, do you have more cells now or when you were a little tiny baby?
Yes.
Well, start off as one tiny cell, another little cell going to.
Yes. So are there more cells in your body now or when you were a baby?
Well, you
Yes.
grow cells, and when the cells duplicate, and then until you get to a certain number, they're done. And you're always losing cells constantly.
So you haven't answered the question. Are there more cells as an adultish body or as a baby?
Yes.
More. Your question is meant to be less. More.
More as an adult?
More as an adult.
Yeah. And the reason is cells have a maximum size. Right? You don't just like have a collection of cells and then go, oh, I need to get bigger, so therefore my cells get bigger. Right? That's not what happens. Instead, you have your collection of cells, and if you need to get bigger, you just make more cells. Right? So why do your cells have a limit to how big they can get? Do you remember this? It was touched on that one Amoeba Sisters video we watched the other day.
I remember this from our first Get video. They have to use oxygen.
They have to use oxygen.
In a certain way.
So how does oxygen get into the cell? It diffuses, right?
Yes. And if it's
And it
too big-
diffuses from the surface of the cell.
Yeah, that was it.
But it uses oxygen based on its like overall cellular metabolism, right? Based on its overall volume. So there's a difference between the surface area and the volume. So we just kind of touch on that real quick. And so we're going to keep this super simple. We're going to do some math, but we'll use a cube because cubes are really easy to do the surface area for.
If you read it, square cube.
So it's not too dreaded. So we have a little cube versus a big cube, and it's not even going to be that much bigger. Let's make it twice as big. Okay, so this surface area, we're not even going to use cells. We'll just use like little imaginary cubes here. We'll say that's 1 centimeter by 1 centimeter by 1 centimeter, right? It's a cube. This one's twice as big. We'll say it's 2 centimeters by 2 centimeters by 2 centimeters. So if you have a cube that is 1 by 1 by 1, what is the total surface area of that cube?
6.
6, right? Because each face is 1 by 1. 1 times 1 is 1 times 6 is 6. That's a good touch. Okay. So this one has a surface area of 6. And then the volume. What's the volume of a 1 by 1 by 1 cube?
1.
1 times 1 times 1 is 1. Okay, what about the cube that is 2 by 2 by 2? What's the surface area of that cube?
12.
Well, what is each face? One of the faces is 2 by 2, right? What's 2 times 2?
I was thinking of it wrong. 4.
4. What's 4 times, how many faces are there?
6.
6. What's 4 times 6? 24, yep. So the surface area is 24. We'll do square units. And then the volume is cubic. What is the volume of a 2 by 2 by 2 cube? Eight. 2 times 2 times 2 is 8. Yep. Cool. So what is the ratio? What is the surface area over the volume? So do that calculation real quick. What's the surface area divided by the volume? So we're off of what is that?
24 weight,
So that one's 6. And then this one, 24 divided by 8 is? 3. Yep. So the smaller you are, the bigger your surface area to volume is. So that's the whole reason cells have a maximum size. They can't just get bigger and bigger and bigger and bigger, because eventually their volume will get so big that they just don't have enough surface area to properly function. So that's the reason why cells divide. So we're going to talk about cell division. And in fact, cells have kind of a life cycle, if you will. So we're going to make it as a big circle. Yeah, I'll do it as a circle. Do 3 different sections here. Okay. Most of the time, cells spend in what's called interphase. Right? Inter means like between, so they're between phases. And interphase just means that it just hangs out as this nice little single cell. It's one cell, one nucleus, just hanging out doing its thing. We'll get into more detail there in a sec. And then there's 2 other main sections. This is when a cell begins to divide. It is called mitosis. You may have heard of it before. And then finally, when it does the actual division, in fact, I'm going to draw one arrow going out this way, one arrow going around again. This is called cytokinesis. Cyto meaning cytoplasm, kinesis meaning physical movement, or in this case, splitting. So the cell will physically split. So here's our daughter cell and our other daughter cell. Should have drawn them the same size. Mitosis. We have 2, at the end of mitosis, we have 2 separate nuclei. And at the beginning of mitosis, we have just one cell with one nucleus. So we're actually going to jump into interphase here. And there are 3 sections to interphase. You see the 3 arrows I drew? The first one here on the right, we're going to call it G1. And the second one is called S. And finally, we have G2. And you remember G1 because G stands for growth, so that's why you have G1 and G2. The cell is growing, and then we also got the S phase. Wait, double check. I don't want to get wrong. G1 phase is when things like the organelles sorry. Thought I had this down. The book doesn't say. What does the book say? I'm trying to remember when the organelles double. Sweet, thank you. Yeah, okay, well, we won't worry about it. We'll just note that G1 is a phase of growth. The S phase, however, this is when the DNA is doubled. So one of the trickiest parts about this cell dividing is it needs 2 nuclei, right? And it needs to have all of the DNA that the parent cell had. So the S phase is when the DNA is doubled. And then G2, more growth happens. Yeah, book doesn't say when new organelles are grown, so I won't worry about it. We'll just put more growth. Incidentally, there's another phase that happens. Do all of the cells in your body divide and make copies? No, they don't, right? You'd be a hideous flesh beast. You'd be a giant blob of cancer, right? So usually this only occurs in cells that need to replicate and need to make copies of themselves. When a cell wants to go off and just do its own thing and never replicate again, right? Like a nerve cell in your brain or something like that. It's like, hey, I'm done making babies. I'm just gonna be myself, do my function. It enters what we call G0. Which is, it stands for gap actually, but I remember it through growth because this is part of the growth phase where it gets bigger. So no replication. Cool. So most actually, over 90% of your cells are actually hanging out in this G0 phase, where they don't have to replicate. You do have places though where you're just constantly churning out new cells. Where is that? Any ideas? Bone marrow? Yeah, you're actually, so your blood cells, red blood cells, they only last 90 days. Or maybe 120 days, 90 to 120 days for a red blood cell, right? So you're constantly making new blood cells, right? You're constantly making new immune cells. That's one place. Anywhere else in your body where you're like losing cells all the time? Brain, not so much. Like nerve cells are like made and then just kind of like hang out. You don't make a lot of new ones. But you've heard of you must have heard that you're like sloughing off cells, right, off the surface of your body, right? All the time. Yeah, you're losing actually scary amounts of cells just off your skin. So skin cells, you're constantly making new skin cells. Same deal with like the lining of your gut and intestines and stuff. Like you're making new cells to replace the ones, because as your food goes through it, like scrapes along and damages tissue and stuff. So you're constantly making new ones there. Cool. So we're going to zoom in a little bit on this mitosis part. This is the complicated part. Remember, G1 is just growing, G2 is just growing. S phase, the DNA is doubled. So we'll talk about that just a little bit. So here's our cell. And it has a nucleus, right? I'm going to draw in the nucleus pretty big. And most of the time, your DNA is all spread out. It's unwound, it's unpacked, it's being used. Your DNA is in separate chunks, right? You have 46 separate chunks. They're called chromosomes. But when it's all spread out and being used, this form of DNA is called chromatin. So you'll have like multiple chromosomes, but they're spread out, they're being used, translated into proteins, things like that. And then one of the first things when you start mitosis is that chromatin turns into what are called chromatids. So this is how it's normally hanging out in like interphase. Oops. Ignore that. Don't draw the nucleus. When mitosis starts, the nucleus actually breaks up. And the chromatin condenses. So each chromosome actually condenses down into what are called chromatids. And that gives you kind of these familiar X shapes. There's one more organelle we need to cover, and that is a centriole. I don't think we covered this in our organelle list, but it was in the books list. I never had to do it, so Remember, there's 2 of everything at this point. And these centrioles, they act as like physical centers to the cell. So what they do during this first step of mitosis is they actually separate. One goes on one side, and the other one goes on the other side. And this first step of mitosis is called prophase. Here, pro doesn't mean positive, it means early, right? I should label these. These are called centrioles. Okay, after prophase is metaphase. And in metaphase, again, there's no nuclear envelope, but all of those chromatids, they line up down the middle. Like this. And tomorrow we'll do a lab. You'll actually be able to see cells that are lined up like this. The centrioles are at opposite ends. And in fact, the centrioles have these fibers that connect to each chromatid. And then in the next phase, they start getting pulled apart. Next phase is anaphase. Centriole, centriole, centriole. But the chromatids are getting pulled apart from each other. And remember, this is DNA that's been doubled, so it's actually 2 chromatids together. And each one is identical to the other one, right? So the cell made a copy of its DNA, and these are 2 identical copies. And then those identical copies are going to separate. So there's this big microtubule. It's a big chemical rope type thing that connects to these, and then they get pulled apart from each other during anaphase. And then they get pulled so far apart that they can recondense back into nuclei. So that nuclear envelope reforms. And the chromatids turn back into chromatin. This step is called telophase. So technically there's only 4 phases to mitosis. There's prophase, where the envelope separates. and the chromatids form. There's metaphase, where the sister chromatids line up down the middle. There's anaphase, when the chromatids begin to separate. And then there's telophase, where the nucleus reforms and the chromatin reappears. But after mitosis, the final step is cytokinesis. I'm going to draw it like that, kind of. Didn't leave myself enough room. But the point is, that's where the actual pinching off and dividing of the cytoplasm itself occurs. Cytokinesis. At the end, we end up with two daughter cells. They're usually considered daughter cells. I don't know why there's no son cells. Okay, so what I want to do is we're going to take a little break, but here's what you're going to do. Here's your assignment. We need some way to remember prophase, metaphase, anaphase, telophase. So we're going to come up with our own personal mnemonic device. What was that? PMAT. PMAT's a good way. PMAT is a good way to remember it. But we can get a little more creative, right? So I'll give you guys maybe 4 minutes. I want everyone to come up with their own, what's the right term? Anagram, mnemonic device. So how are we going to remember the letters P, M, A, T in a row? Like, please make anyone talk or something like that, but be creative about it.
I made up PMAT. Completely original. In the moment, it was entirely creative.
So come up with 4 words in a row. One that starts with P, one that starts with M, one that starts with A, one that starts with T, that conveys something. You can do this. Blaze, I'm gonna give you 4 minutes.
Alright.
And then I want your creation at the end. It has to be school appropriate, but the more interesting it is, the easier it is to remember. Stay on. 1 minute left. You're going to share this with the class. Okay, Blaze, we'll start with you. What'd you come up with?
Alright, so for P I put pterodactyl.
Pterodactyl. And
Pterodactyls might attack T-Rex. I just thought it was
pterodactyls might attack T-Rexes.
I just thought it was funny because pterodactyl does technically start with P, but you know.
Pterodactyls might attack T-Rexes. I love it, actually.
Also, please
That's-
misuse all time bombs.
That's hilarious. Have you heard the there's a children's book that's like,
Oh yeah, they're like, isn't it like the worst alphabet book ever?
yeah, the worst alphabet book ever, where they like all pick words that like start with one letter but then have a different sound, right?
Also, please misuse all time bombs.
Oh no, don't take too many good ones. Brandon, next.
People matter at train stations.
People matter at train stations.
Why specifically train stations?
I think, what'd you get?
Freddie met on today.
What was it?
Freddie met on today.
What is that? Patty met on today? What does that mean?
Nothing.
On today? Like what like AHN?
No, ANN.
ANN. And today. Okay, back table. Madeline, go ahead and start. Wait, what was it?
I didn't get it.
Oh, no, okay, we'll give you more time. We'll wait. It doesn't have to be an award-winning version, but PMAT, 4 words in a row. Make a little sentence.
I can't do
We'll come back to you. Kaylee?
I don't feel it.
Oh, we'll give you, okay, we'll give a couple more minutes, let you guys come up with one.
Daring today, aren't we?
I thought I gave everyone enough time, sorry. How's your soda?
He sucks.
Is
I hate it.
it too strong? Is it weird flavor? Can you taste the yeast?
I can taste the yeast, but it doesn't taste good.
Yeah. I mean, yeah, no, it doesn't taste good. It tastes like sour, right?
People drink this regularly.
Do they?
Well, add that more alcohol side of things, yes, but.
You're not tasting the alcohol, or the alcohol's really minor. It's mostly the yeast, the yeasty flavor.
Yeast is better in solid form.
It's better in bread,
Yeah.
like a good like sourdough bread.
Did I just go horribly wrong with mine? What does everyone else taste like?
I haven't tasted mine. I'm not,
Taste it.
I'm not gonna taste it.
Oh, no.
Oh, you're not even gonna taste it?
It smells.
I'll taste it. What did you put in it? Just regular root beer?
Yeah, just regular root beer.
Then drink it. Otherwise, you go to the weenie house.
Here, bring it here. Bring it here. I want to try it.
You don't want to be part of weenie house.
Oh, you are gonna drink it.
Not that bad, but just real bland.
Oh, you're not gonna drink yours?
Sour aftertaste.
It's a bit strong. It's no, it smells.
Yeah, we did new seats. You are. Where you at? You're right up front. The place.
Why does it taste better than bread?
Does it taste better than bread?
Not better than bread. Why does bread taste better?
Why
than
does
this?
bread taste
Same
better? I don't
thing,
know.
Just different
Oh, look how fizzy it is. It's so carbonated.
You know, you just
Smells like root beer and yeast.
funny how that works.
It's about right.
Does it taste awful?
No, it's not
Smell
awful.
like you would-
There is no strawberry. I added just
Yeah, it
enough
just tastes
strawberry
like
that
green
makes
yeast.
it. There is no strawberry in this.
The
That's a
yeast,
lie.
the yeast ate the strawberry.
That's not how that works, first of all. Second, where's the strawberry? I was promised root beer. I got hell on a stick.
You got hell of a
I don't know
bottom.
where the stick part comes from, but it's there.
Madeline, you ready? All right, let's hear it.
Please make a taco.
Please, what?
Make a taco.
Make a taco. I love it. Kaylee, what'd you come up with?
Misuse, please misuse all time bombs.
Please misuse all time
I
bombs.
came up with two. I can give one away.
Kennedy or Diana?
I just took the beginning couple words and put them together, so pro, meta, and
Wait, you just did what?
see, the beginning part of the word, I just took that and then that.
Pro, meta, Anna, Telo.
That was good. I couldn't think of anything else.
Sounds like something like akin to supercalifragilistic.
Okay, well, we'll let that one slide because it's correct, yeah.
People made applesauce.
People made applesauce today? Nice. All right, Kaylee, what'd you come up with?
I'm still thinking, I don't know.
Okay, we're gonna come back to you. All right? Over here. What'd you guys come up with?
People make another Chernobyl.
People make what?
Another Chernobyl.
Another
Chernobyl.
Chernobyl. Another T-Robe-Noble. Close enough. Chernobyl is C, yeah. Zion, Nevada, what did you guys come up with? Okay, shh, 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 shh. Sorry, what was it?
I got the exact same as you did. Please make taco.
Wait, please make what?
A taco.
Really?
Yeah.
How do you guys independently come up with the taco thing?
Because tacos taste good.
Somebody, maybe you both work at Taco Bell or something. Please make a taco. Please make a taco. What'd you come up with?
People make all trucks.
People make all trucks?
I mean, not
At
wrong.
least they're not tacos.
We do make all chocolate.
People make anything? Sorry, what, please?
Policemen are tired.
Policemen are tired. Oh, that's a cool one. That's different at least than please make. I'm gonna give you 30 seconds. Come up with a mnemonic device to remember PMAT.
Platypus mostly.
No, 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 no. Jalen's gonna come up with it.
I'm coming up with my own.
I've already made like 5.
Plant my apple tree. I love it.
Patty made a toy.
Patty made a toy. Love it. All right, sweet. We got there.
Mostly a salt turkey.
PMAT. If you can remember PMAT, you can remember prophase, metaphase, anaphase, telophase. Or at least, if I give them to you in a test, you can put them in the right order, right?
Platypus mostly attack turkeys.
Platypus mostly. You could just roll these out all day, couldn't you? Usually with animals. All right, sweet. We're gonna watch a video. We got 10 minutes left. 25? I'm trying to get out of here. Oh, 40. Oh, we got lots of time. Okay. Yeah. What's the what for today? Today is a Thursday schedule.